free radicals and how the reactive oxygen species interact in the body and how they get generated, is why they get generated. That is one of the big questions. And antioxidant defense system in the body, how they maintain, how they substantiate the clinical significance, means it is a condition when they come across, and it is a condition where the free radical generation is good. What is the condition where the free radical generation is bad? Then we will be trying to cover these first two objectives today and try to understand them in detail. And the last objective of the learning, that is the role of oxidative stress in cancer, diabetes, atherosclerosis, and so other diseases for that. So that is an unending list. That is your translation research of that basic understanding of the biochemistry. And maybe today, this is a class which can recall you why it is not a chemical biology you are studying, why you are studying the biochemistry, why the chemistry is important to understand the human body, though expected thing is the reverse. You have to be the clinician, you have to be the surgeon. And why we have not given importance to the biology, why we have given importance to the chemistry. Maybe today's class you can understand that is also one of the objectives for me to prepare the class such a way, so you should come across to this. Now, I will not talk about, all the things. This only, as today's class is just your revision of your 12th knowledge, that what are the free radicals, how they generate, and as you know that it is actually it is very active molecules because it's unpaired and not stable, having high energy, and try to move and bind to with any of the other available species for that. So here question come for that, then where it is happening in the nature, is fine, which is the other students are studying, or the other in the biologist is studying how they are reacting to the plant, reacting to the environment, or reacting to the animals. But our major question is that what is happening this free radical in the body? And obviously, one of the most abundant molecule which we need for survival is water and oxygen. So these are the two places where this free radical can go and can initiate a free oxygen species or free nitric oxide species or free H2O2 molecules. So these are the three places where this chemical reaction is important for that. And as you show that is again I just took it from your basic book, that out of that is the 8 saturation stage. If one goes out, that's making it more acceptable for that. So one single pair of electrons which will go out, and you see it's having 7, and it is free to get bind to any other electron. And these are some of the common free radicals in our body, or we come across, which are in vitro and in vivo. So till this, it is a common for all. It is either, as we say, that we cannot, the present situation that we see in this present epidemic of the COVID virus, you see that we have to be rethinking about the approach as taking the whole biology as a whole. It is not the virus, it is not the bacteria, it is not the plant, it is not the animals, but these all species can react in the same way, and with not only the human, it can affect the other also. So knowing understanding of the biology, what it means, that if these molecules, you can utilize them to harm a virus or a bacteria or other unwanted species, it's fine. But if it harms the human itself, then it is a bad. So it is a basic definition of good and bad. So nothing is good according to me, nothing is bad, but how judiciary we are using these molecules, that is most important. So here, I just want to recall, so superoxide hydroxyl radicals, hydrogen peroxide, oxygen, and hydroxyl radicals. These are the some of these species, and many more, which I am not talking. Why I am not talking about the many more? Because all the xenobiotic can convert into the free radicals. All the proteins can be affected with this, some and other drugs. And what is exactly happening about its generation as a free radical, that we can't review in such a short time. But that is a big question for you when you join medicine or you join the pharmacology, to ask that, is that drug can generate the free radicals? If either they generate their own free radical or they generate the known species of the free radicals. Now, it's the same thing I have written over here: free radicals, so superoxide radicals, hydroxyl radicals. This is the list which are the most susceptible for the humans for that. So important characteristics of the reactive free radicals, reactive species oxygen free radicals are: they are steam reactivity, they have very short lifetime, few million seconds to few minutes, generation of new RSS by the chain, and damage to the well. So these are the four important part for that. So that's it is like our nuclear what is the isotope preparation. Once you start bombarding, it can created. So this we are more affected by that, this chain of the reactions, as you say, that once it's started and it is unending. So now the question for us as a clinicians, how to stop it. Second question is that if it is going to damage, so what is the organs it is going to damage? Which is the most first target, second target is this. We have all the answer, you know about it. There's many of the diseases. So for example, in case of the diabetes, it is the case of the retinopathy or various other aspects. I will come when talking about the disease for that. Secondly, one most important aspect for that, ultimately, what is the extent of the damage? It all depends for that. Say, if it's a damage is big, okay, it can be repairable. But if the damage is more, it is not repairable for that. So I have given here that, of course, it is again bookish knowledge, nothing to remember that few minutes, the microseconds, 10 to the power -6, for example, oxygen, very powerful radical, superoxide radical, and that's for that few up to the few minutes up to for that 10 minutes is ozone is quite a bit for that, so you require a longer exposure to stay in the body for the longer time. So, and it is not removed. So this timing shows that how important it to take place the reaction, and unless and until we prevent it in the beforehand, it's not possible to develop any drug to directly control it duration of its generation, the time period of its generation. So that was the major thing why I'm showing the half-life, because half-life is always either when you discuss about the drug or you discuss about whether it's any of the xenobiotic, this is the key molecule. Why today we are worried for that so many of the drugs and xenobiotic enter in the society, in the environment and in our body, because all of us are moving with that amount of the xenobiotic. So the reason is that what is their half-life. So this is another example of the importance for that. So I'm just taking a quick example of that generation of free radicals. You see that's how they can get generated. The electron can go to oxygen and it make it active this negatively charged oxygen, and it gets bind to the hydrogen and H2O2, and ultimately H2O got released, and this is a remedy for that. And the free radicals can be available for that. So other generation of free radical is the NADPH oxidase system. So that is basically the neutrophil, eosinophil, where the NADP is generated and oxidase system and produce superoxide Ni. And you know this is we remember HMP pathway during the glucose metabolism. So HMP pathway, that's glucose GPD, and it's converted into that the help of the NADPH oxidase that takes place, that SOD convert into the free radical hydrochloric acid, and this is this oxygen free radical can attack the bacteria and macrophages and respiratory bursts. So this is that basically the polymorph, that PMN, and is an important site over this free radical generation can help you for the killing. Because what happens, the macrophages ultimately catch hold of your virus, bacteria, or any of the unknown thing, and that can be attacked by the generation of this concise cycle where that HMP shunt pathway help you for that to produce the NADP, to produce the NADPH, and that in turn convert into oxygen to the oxygen free radical. Of course, after that, this oxygen radical directly can go and attack, but and we cannot allow this whole reaction to keep repeating, because after the bacterial kill, it will start killing your own cell. So other line of defenses here with the SOD, that superoxide dismutase, this enzyme comes in the picture, and see how nicely that enzyme is also prepared for that in the body. It's a basic enzyme, SOD and catalase, and how they are converting into that's the inactive molecules, which is an H2O2 for that. Similarly, I've just given some of the SOD. Catalase also worked on the H2O2, convert into water, glutathione peroxidase, and myeloperoxide. So these are the basic free radical things, reactions where it can be get bind for that. Then iron also can act as a Haber-Wieschen reaction. One of the reactions we have iron role, where that oxygen free radical and H2O2 get reacted, and this ultimately producing the hydroxyl radicals, glutathione reductase, NADPH oxidase, and Fenton reactions, very important reactions for that, where the ferrous convert into ferric, and this ultimately can act as a free radical reaction involved in the reactive oxygen species. Fenton reaction and Haber-Wiesch reactions are dependent on the iron. This too is a iron dependent reaction. So generation of why it takes place for that, due to the leak in the electron transport chain in the mitochondria. That is because there is a reason mitochondrial cell wall are so tough. It is having a unique structure in the cell. It is having away from the nucleus, so they have the more space for that its own function. It is a wire. It is, you can say, it is a power generating mechanism. So one of the most important part is that in case it gets leaked from the mitochondria, about 4% of oxygen takes up in the body is converted into free radical. So that is another very important part. But so if we, of course, it never, that's the reason if you are in under meditation or where you are consuming less oxygen, you are helping your body to react or relax with the less amount of the free radical generation. Now, this is a general scenario which we can see. We see for that. So you cannot avoid UV, this radiation, because all through in my life, 40 years, I worked under the radiation, and this is one of the very drawback for us for that. So of course, my exposure to radiation is less, but I'm just thinking about all of you who will join the radiology. You have to be careful, because this free radical generation damages that. Of course, it is a important aspect in the society. We have taken all the steps, but this secret, each secret is sufficient. I'm not going about that data, that how much it generates the free radical and what is the quantity, what is that. But actually, it should be prevented. It is not a, it's a important issue to discuss about its generation in the cigarette smoking, but it is important to how to prevent this. Then, of course, air pollution we cannot overlook, and that part you have already seen. That's how it is, but it is again unavoidable in the civilized fashion for that. Then it is next important is the why. Just a minute, let me, in case anybody calls. So other is of course mitochondria. I have already told it's a generate radical and it can be leaked, and white blood cells. So these are that. But ultimately, what happens? This is the most important part, which is related to many of the disease, particularly the DNA damage, because that gives the best theory. Because even I'm not taking the students who are not attending my tomorrow's class. So this is a key mechanism for generation of a cancer. How to the cancer? So DNA damage is one of the very important aspects how the formation of free radical can get. Of course, these remedies I have already told that what had happened. We have just a free radical enzyme, that SOD and glutathione peroxidate and glutathione redox. So this is the redox cycle. So practically we have done a lot of practical work on it, where that we have seen that how to quench this molecule, what is antioxidant therapy for that to control this type of the mechanism. But this is a $1 million slide, which is, if you can understand this one, that's SOD and glutathione peroxidase and glutathione redox system, which is the major free radical scavenging enzyme, which takes the two wonderful molecules. One is glutathione, and other is NAD. So glutathione is a molecule which is like cyclic AMP and like any other novel molecules which came into the limelight, the very small peptide, but having a wonderful mechanism for that quenching the free radical. So what similarly, as I told you, catalase also. So H2O2 plus catalase will convert into water for that. So these all are the two important beside the glutathione redox system, SOD and catalase are there. Now, this is the one part of the chemical part. Now, when we come to that, some of the basic, why we in general we say that okay, we should take the diet rich in the polyphenols, and which is available in the vegetable fruits and etc. And they contain flavones, isoflavones, stuff like that, and catechin, phenolic acids, and these all are basically act as antioxidant, anti-apoptotic, anti-carcinogenic, and anti. So in other terms, whenever there's any unwanted free radical get generated, try to quench, try to engulf. The same work can be done by the macrophages, but it also have some repercussion, because macrophage is again made of the certain cell wall, other thing, what that free radical take the advantage to get destroying the cell wall itself. But in case we have such quenching molecules, they are the wonderful. Maybe that is one of the plus points that's, as you see in the today also in the remedy, that's people, vegetarians are the consuming the more flavor compounds are less susceptible to the present virus infection for that. That is one of the study recently it has been shown. So this is always good if we can have the anti-inflammatory compound. Beside the, we have two known vitamins, vitamin C and vitamin E, which we will discuss next class. So this basically this is they can damage produced by reactive oxygen is that they can damage the protein, they can damage the lipid oxidation membrane, mitochondrial permeability, and DNA damage, as I told you, cell death and the cancer for that. So this is a key role in the case of reactive oxygen species. So finally, what is the clinical significance of that? If we know, having known that, discussing about that, it's okay. Free radical get generated, there is a system for quenching it. Then how it is what is this clinical significance? So one is chronic inflammations. As you know, the best example, rheumatoid arthritis, for that's acute inflammation, disease in the eye, and for example, retinopathy, cataract for that. So these are some of the first side where that get affected for that. So how that free radical can get directly go and bind and to the respective cell wall and resulted into that, making the disease condition for that. So that is other is perfusion. So that is a very, very important for that. The myocardial ischemia, it happens that it is that your basically that hypoxanthine can convert, that is another source from the ATP. Xanthine oxidase convert into xanthine. But during this process, oxygen can change into the oxygen free radical, and that they convert into that and try to block the reaction of the peroxidase. So this is where that generation of the more free radicals can occur, and xanthine oxidase is the known system where that how to generate the free radical for that. This is under urea cycle. You know the xanthine oxidase. You have already studied about it. Then atherosclerosis and myocardial infarction. That is LDL are deposited under the it should be the endothelial. E is missing. Endothelial cells, and which undergo oxidation of free radical, making the foam cell, and that is ultimately going to block the arteries. So antioxidant offers some protective effect, and carcinogenesis, as already I told, this free radical produce DNA damage, and accumulated damage lead to somatic mutation and malignancy. Then finally, that's aging. That is another gray area where that's aging, and you know that if you can somehow you can stop the free radical generation by whatever theory we are saying for the meditation or with the yogic science and whatever that they say for that. Somehow you can stop the free radical generation. It is resulted into decrease in the aging process, and basically this biological aging due to accumulation of reacting oxygen metabolite with the metabolites, and such diseases are one of the outfall, that is a Parkinson, Alzheimer, dementia, multiple sclerosis, and is the ultimately cumulative effect of the free radical generation. So here, in a nutshell, it is my take-home message for all of you that free radical can cause the oxidative stress via, as I told you, the generation of the free radical, as I have shown you for oxygen, nitrogen oxide, xanthine oxidase. So many molecules can be involved in that. It resulted into affecting brain, heart, kidney, skin, multi-organs, joints, immune system, blood, lung, and that. And there is a various evidences you can take for that, and diseases I have just written here, like in the case of the lung, asthma, COPD, allergies, and eyes, I told you the retinal degeneration, cataract is one of the common for that. Immune system, this is one which is very widely studied, and still you can do for that, because some of the easy way how the free radical generation is changing the antibodies functions. That is became one of the important aspect in the case of the arthritis for that. So, having studied about their clinical application, generation of free radicals, and the parameter of this possible intervention and their clinical application. So, how this, we just see that what is the mechanism, how it starts the generation of the lipid peroxidation, because throughout my talk I have told they are going to affect that your cell wall. So ultimately, what is the cell wall destruction? That mechanism is through the lipid peroxidation. So it has first phase is that initiation phase, where the polyunsaturated fatty acids present in your cell membranes are get affected, and by this free radical bombarding, and this ultimately generation of the PUFA radicals or the lipid peroxidation radical, and it is a chain of the reaction, keep going from one cell to other for that. So this is one peroxidation of poly and PUFA that's ultimately gets free radical get to bind, and PUFA free radical can generate. This is the chemistry behind it for that, how we generate. And this is here again, I'll show you all these R radicals are the free. They can metallize and other things can generate for that. So that is initiation. It's a propagation, you know, once it gets reacted. And then see, they keep changing it for that. And carbon anti-radicals rapidly react with the molecular oxygen forming peroxide radicals, which will take the another polyunsaturated lipid molecules, and you see the reaction keep going on. I told you this is a chain reaction. This would lead to continuous production of hydroperoxide and consumption of PUFA, and one free radical generate another free radical, as I told you already. I have told you for that in the case of nuclear reactions, for the chain reaction. Ultimately, it is called as a death kiss by the free radicals. So finally, of course, there must be some way inside in the body. It is not that even when we have so many harmful effects, and without intervention with the clinical, without intervention by the physician also, many of the diseases get checked, and it is one here, it is the termination phase, unchecked till the peroxyl radical react with another peroxyl radical to form an inactive product. So this also keep happening inside the, particularly in the macrophages killing system for that. So others, we can prevent it by the use of antioxidant, as I told you, catalase, glutathione peroxidase, ethylene diamine, EDTA, and these will inhibit the initial production for that. And chain breaking also takes place with the presence of many of the vitamins, and these two vitamins I told you, the ascorbic acid and the alpha tocopherol. So they have the capacity to quench these free radicals over here and can make it as an inactive product for that. So basically, the most effective naturally occurring vitamin E is the most effective naturally occurring chain breaking antioxidants in tissue for that. So vitamin E consumption is very, very important, and only traces of tocopherol is required to protect considerable amount of polyunsaturated fat, and say 1 tocopherol molecules per 1,000 lipid molecules. So it is a very advantageous for that. So vitamin E supplementation is very, very important in such cases for that. It decreases the aging also. So acting an antioxidant, alpha tocopherol is consumed, and it is also replenished the daily dietary supply. So biomarker, now you may raise this important question for that. So, okay, then how will come to know that we are suffering from oxidative distress? So there is a various established parameters for that. One is the malondihyde and hydroxy non-amyl molecules, acrolein, there is propineline. So these all molecules are a marker of the free radical generation. So once you can measure, you can just, even you can do that total reducing capacity in the cell. there is an available method where they take care of all the glutathione and other molecules together, reducing capacity of the cell. It shows that how susceptible you are to that's oxidative stress and how much power you have. And lipid peroxidation is a measurement MDA, that is actually thiobarbitic acid in the lab we use to having this basically the MDA estimation for that. So that gives you that idea about that. What is lipid peroxidative stress present in your blood? This test you can do in the laboratory very easily. Then, of course, this protein cleavage, cross-linking inhibition, enzyme activity, alter immunogenicity is very important. If you take that, actually, if you take that some of the known protein molecule and look for that, how much they damage, the degree of damage for that. This is this cross-linking inhibition. This part is very, very important in the case of the allergy and in the case of arthritis and other places where the directly is the protein cleavage takes place due to the free radical. And then 8-hydroxy, of course, 8-isopropyl. This I just mentioned is prostaglandins. So PGF2 is a stable prostaglandin that stimulates the contraction of uterine. So their breakdown into that 8-hydroxy PGE2A is the most reliable assessment of oxidative stress, new risk factor for coronary disease. So this one, very recently it has been added 8-iso PGE2 labels further in the case of the particularly in the case of coronary diseases, and gives an early marker for that. Other markers are glutathione, as I told, glutathione disulfides, and there's nitrated proteins in diseases, carbonated proteins, mostly that is carbonated due to the lysine, arginine, proline, threonine for that, and DNA break, that 8-hydroxy guanosine. This is one of the most common for that. And beside this, you can do that leader assay also of your DNA. Just do the electrophoresis, and you can see how many breaks of the DNA you can get from that. So what is the remedy? Remedy is that this is a vitamin E, as I told you, quenching compound, vitamin C, cerebroplasmin, caffeine, cysteine, glutathione, vitamin A, and beta-carotene. So these are the one of the important. Of course, cerebroplasmin acts as an antioxidant in extracellular fluid, one of the most important part for that. And caffeine also help you for the dietary source. And of course, vitamin E and C is one of the most important one in the aqueous phase, and other is in the lipid phase for that. So I think we have finished with all, and whatever that's query in the next class, we will just add on to it, taking some of the diseases for that, but most of the diseases I have given you today, a framework to think about it, and tomorrow we can be discussed about more about the disease etiology. So 2 textbooks I have gone through. One textbook of biochemistry by Vasudevan, 9th edition. So they have described most of the thing, and it is there in the internet also. You can share it. And Harper Illustrated Biochemistry, of course, gold book, which is a bit difficult, but it's a long run. I think this is the best book